Well, hi everyone. It's good to see you. Let's get started. I'm Will Alexander, dog show judge, educator, and retired handler, and also the host of Dog Show Tips. And on behalf of Purina Pro Plan and Paw Print Genetics by Neogen, I would like to welcome everyone tonight to the webinar on preparing on large breed genetics and reproductions. We have a great presentation in store for you tonight, and I'm glad you can all make it. First up is that's me. I already gave you my quick bio. And here we have Dr. Westra. Dr. Westra has served as a professional service veterinarian for Neogen since 2020. His position with Neogen Genomics blends a 10-year career as a biologist with 12 years of clinical veterinary medicine. He is passionate about science, education, and helping dogs live their best lives. And Dr. Greer is a renowned veterinarian and lawyer whose life is dedicated to improving the health and well-being of pets. She has extensive experience in breeding and raising show dogs and has written the book Canine Reproduction, Neonate and Neonatology, a practical guide for veterinarians, veterinary staff, and breeders, and your pandemic puppies. Our agenda tonight, we'll kick it off with Dr. Wester's presentation on large dog breed genetics, a quick message from Paw Print Genetics by Neogen, followed by Dr. Greer's presentation on large dog reproduction considerations. Then a quick message from ProPlan. We'll have a 5-minute break. We'll head right into a 30-minute Q&A to wrap up the evening. A little bit of housekeeping before I turn it over to Dr. Westra. We're a big group tonight, so we're going to keep everyone on mute during the presentation. Please enter your questions from the Q&A period anytime into the Q&A box. It's a 30-minute Q&A period, and we'll get to as many questions as we can. Please take a moment to note the difference between the Q&A box and the chat box. We'll be taking questions for our presenters exclusively from the Q&A box, but feel free to use the chat box to connect with each other during the presentation. Finally, the doctor would like to ask you to keep your questions to the general topic they'll cover today versus specific ones to your own dogs. With that, I'll turn it over to you, Dr. Westra.
Thank you, Will. Appreciate the introduction. I just want to welcome you all tonight. Tonight, we're going to be talking about genetic concerns of the large breeds. But before I get into the material that I presented, I got a question for all of you. And I don't know if I'll be able to see the chat here in presentation mode. So I guess I just want to start off with, what do you think the biggest concern with large breed dogs are? We're talking about what do you think their biggest health concerns are? Bloat, longevity, hearts, okay, joint health, digestion, yep, hip dysplasia, cardio, hips and elbows. All right. So let's see if I can minimize the chat now. So those are all excellent questions, and hopefully I'll present some information for you tonight that may give you some greater understanding into how the field of genetics is addressing some of those specific concerns. So we're looking at a number of diseases and traits of interest in the large breed dog. And so these are just some of the things that I know that there is current active research on. And so I've got musculoskeletal conditions, which were some of you have mentioned. A big one that is always of concern for large dogs is hip dysplasia. And kind of part and parcel with hip dysplasia is osteoarthritis. And then there is some significant research being done into ligament injuries, mainly the cranial cruciate issue. And then, of course, with large breeds, we're looking at behavior. It's kind of one of the nuances when we're dealing with a large animal. We want them to have the appropriate behavior, and we're wondering if genetics may provide some insight in how we can continue to, keep these companion animals. And of course, there's a lot of interest in just the morphometrics, and that is the genetics of size and shape. There is considerable concern in allergies, which affects many of these breeds of dogs, as well as cancer, which kind of speaks more towards the longevity concern that was raised. And then nutrition, like these are big dogs and we have to feed them a lot. And I'm sure Dr. Greer is going to have some insights and some helpful tips in this. But I wanted to look specifically into some of the genetics of it. Now, I can't cover all of these topics. That would take, probably a whole month-long seminar. But I would like to focus in on two of these that I've highlighted in red, and that being hip dysplasia and obesity. So to go through this, we're going to have to cover a lot of science, and we're going to do this in about a half an hour. So I apologize as I move fairly quickly through it. So if there's something that I go through it doesn't quite make sense or you don't quite pick up on the relevancy or the importance of it, feel free to ask us in the chat afterwards. I'd be happy to try to explain this in greater detail. But I'm going to try to address some of these questions. And first of all, I'm going to be presenting a lot of genetic concepts that apply to how we're doing genetic testing today. And the first is, how is the sequencing of the future, how is sequencing the future of genetic testing? And then I'm going to cover a little bit about a very unique type of mutation called a structural variant and how it applies to both hip dysplasia and obesity. And then, again, I want to focus in specifically in, are big dogs destined for bad hips? And what do we know about the genetics of hip dysplasia? And then finally, I'd like to wrap it up with things is that why do dogs get chonky? And try to see if I can understand some of the, explain some of the genetics behind a very, I don't, I almost can't avoid saying the pun, big concern for these dogs. And first of all, I want to just talk a little bit about size. Dogs are absolutely genetically amazing. The domestic dog is probably the most variable sized mammalian species on Earth, displaying at least a 40-fold difference between the smallest of the breeds and the largest of the breeds. And this speaks a lot to the genetic diversity within the species. And at the same time, it shows that how our domestication and how our focused and captive dog breeding programs has really created a very diverse presentation or phenotype of the genetic underlyings of this species. So when we talk about size, this presentation that we're giving tonight is basically the counterpart to the presentation that we gave last fall, in which we talked about small breed dogs. And I presented this information in a little bit more detail. But the interesting thing is when we talk about dogs, we are currently aware of about 20 different genes that affect the size of the dog. And then of those 20, we know that, or we suspect that one of those genes contributes to about 15% of the overall size of the dog. Now, to contrast this, if you look at humans, height and size is thought to be controlled by thousands of genes. Now, we've been looking at human genetics much more intensely for a longer period of time. So we know a considerably greater body of knowledge when it comes to human genomics. And then of those thousands of genes, the greatest known contributor is responsible for less than 0.5%. And so you can see a huge dichotomy between the genetics of size in dogs versus the genetics of size in humans. So now, in order to get through some of the topics that I want to cover, I kind of got to bring you all up to speed on where genetic testing is. And so I'd like to introduce you to something called a single nucleotide polymorphism. And that's a mouthful, and we don't want to say that any more than we have to. So we're just going to refer to this as a SNP, SNP SNP. And what a SNP is it effectively is a single variation in one of the nucleotides. And when I'm talking about nucleotides, I'm talking about those base pairs, adenine, thymine, cytosine, and guanine, the ACTG. As you can see in the image that's associated with here, each one of these represents a base pair that make up the sequence in the double helix strand of DNA. Now, when one of those base pairs is switched or there's a deviation, we call that a SNP. And as you can see in the thing I've highlighted, that the GC has been reversed from the image on the top to the image on the bottom. Now, when one of these SNPs or variations in nucleotides happens in at least 1% of the population, these are very useful, and they are the foundation for how we go about looking for and detecting the genetic mutations in the breeds of dog. The way I guess that you can look at that is that these SNPs are effectively like a thumbtack in the map. They establish a series of genetic markers which tell us you are here. They allow us to map the genome so that we understand where genes are found on what chromosomes. And they also help us locate specific genes and specific mutations. And if we have enough SNPs and a large enough database, we can use these to create a genetic fingerprint or a profile. So this is all based on the SNP basis of testing. Now, I present this because now we're moving into something different. Genetic testing has significantly evolved, and now we're talking about sequencing. So SNPs basically represent where technology is today. Where we're moving towards is sequencing, and sequencing is just that. We're not looking for an individual variation in a single nucleotide. Now we're looking at the entire sequence, the T, C, T, G, A, C, that make up each of the base pairs in the double helix as it gets wound up into the chromosomes. Now, the advantage to sequencing is we're not looking for a pinpoint. Now we're looking at the whole read, and that it provides the potential to more accurately assess the genetic potential of an organism. So now we can actually say, let's look more at what the sequence is, and that will give us more information to fully explore the genome. So when we compare and contrast, when I say array, array is the technology that we use to detect the SNP. So we're going to compare array technology to sequencing technology. And array technology assesses a known sequence variation. We're looking for that single nucleotide variation that makes up the SNP. Versus sequencing, we determine the entire DNA sequence. And so we get a much, much broader picture. And in order to detect the SNP with the array, we have to have prior knowledge of the DNA variants. And ironically, the way we discover SNPs is through using sequencing, which is, I will go into greater detail how we use sequencing to find the SNPs. But the advantage is when we move to sequencing, we don't need to have this kind of prepared, like, let's look here and see if we can find the SNPs in this region. We just basically are going to be able to sequence the DNA and see what is. Now, the advantage to using SNP technology, and the reason why it is the basis of commercial testing today, it's less expensive. And it comes down to cost and efficiency, is that in today's technology, it's much easier to find these SNPs than it is to do sequencing, but that's changing. We're developing much, much more efficient ways of doing sequencing. And then I hate to say it because it's almost a broken record these days. With advanced computing, dare I say AI, where the ability for us to interpret sequencing is becoming much, much more feasible. Because when we're looking at that whole sequence of DNA, we are going to produce huge amounts of data. And so it requires a certain amount of effort to sift through all that data and find the really interesting things. And that's why SNPs are a little bit more advantageous at this point in time. Now, the thing is that when we use an array chip or something of that nature, it's very challenging to basically bring new tests online. We basically have to reconfigure how we run the test in order to add the new mutations. The neat thing about sequencing is that once we have the sequence, we can basically go back and reassess the data. And we have greater flexibility when it comes to evaluating new and exciting mutations. And the thing too is that with array, we don't use it to discover new variants. But with sequencing, that's the technology that we use to basically assess the DNA and see what's there. And so the one thing to keep in mind is that I don't want to stress that sequencing is something new. We've been able to do sequencing for a number of years now. But what's different is that our ability to do it efficiently and in a more timely manner is greatly changing, which is why hopefully in the not too distant future, sequencing is going to be the basis for all commercial genetic testing that we're going to offer to you. All right, so I went into a great deal about SNPs and sequencing. Now I got to introduce two more scientific topics to you: monogenic versus polygenic traits. And this, we're going to take this and we're going to look at what is the presentation of the genes in the dogs that we're working with. So when we think of a monogenic trait, think one gene equals one trait. The one trait is the phenotype, or what we see based upon the genotype, or what is in the DNA of the animal. And so a great example of this is when we look at coat. The mutation for the FGF5 gene produces whether a dog is going to be short-haired or long-haired. Now, long-haired is recessive to short hair. So a dog has to carry two copies of the mutation that produces long hair, like our collie in the picture below. Its genotype is LHLH, two copies of the mutation, and therefore it presents with the phenotype of long hair. And we contrast that with a dog that has the genotype of SHSH, meaning it does not have any copies of the mutation, or it only has one copy of the mutation, and we see a short-haired dog. Now, contrast this with polygenic. Think mono one, poly many. Polygenic traits are things that are influenced by many, many different genes, as well as many different factors that may not be genetic in origin. And we can see a range of phenotypes when it's based upon polygenic, based upon how all the genes work together. And like I said, these are often influenced by environmental factors. An example of this that you would be familiar with is in coat shedding. Now, there are many things that determine how much an individual dog will actually shed. But what we understand genetically is that a mutation in the MC5R gene has been associated with dogs that shed more. At the same time, because that's not the whole picture, is that you could see a mutation in the RSPO2 gene, which is also the same mutation which produces improper coat or furnishings. And we know that dogs that have improper coat shed more than dogs that have furnishings. And if you look at the chart below, you can see how the two genes, polygenic, work together to create a spectrum of the phenotype of shedding that we look at. Now, let's take SNPs, sequencing, monogenic, and polygenic, and boil this down to something that affects our large breed dogs, and that's canine hip dysplasia. And the reason why I use this is that this is a truly significant issue for large breed dogs. It was first described in 1935 and has been the subject of intense investigation for decades, and is probably one of the most prevalent, meaning it affects the most dogs of all the inherited conditions. Okay. Now, we know that there are many different etiologies, or where does hip dysplasia come from? And so we would describe hip dysplasia as a polygenic disorder with both environmental and genetic components. Now, we know that this is significant because in 1966, the OFA started out with its initial mission was to provide radiographic evaluation, data management, and genetic counseling to eliminate canine hip dysplasia. And although this has been an incredibly valiant effort, it hasn't really gone the way that they thought. And that's because hip dysplasia is not a simple monogenetic. You breed like to like, you should get more like. And unfortunately, that's not the case when it comes to hip dysplasia. We all know that we can breed a dog with excellent hips to excellent hips, and then we just get that one bad apple. And what we're learning now is that because this is a polygenic trait, meaning multiple genes, as we look for more and more reasons, we can start to compile these into a better genetic understanding of what actually causes the inheritability of hip dysplasia. Now, I'm not trying to throw OFA under the bus. I think that they've done an excellent job, and they have become a repository and a clearinghouse for information when it comes to dogs, pedigrees, and health information, which I really appreciate, which is helping breeds across the spectrum of health issues, not just hip dysplasia. So let's look at why we can't have a genetic test for hip dysplasia. As we've talked about, this is a polygenic issue, but let's think about all the different ways that a dog can develop hip dysplasia. And that could be that we could see changes in the alignment between the femur and the hip, or the acetabulum where the hip resides. If these bones don't align correctly, you're going to see the development of osteoarthritis, which will eventually lead to some kind of dysplastic condition. Now, likewise, it can also be not only alignment, but the cartilage that covers the femur and lines the acetabulum, which is the cup and the ball and socket joint. If the cartilage is bad, this also is going to lead to a dysplastic hip. And finally, the ligaments, which hold the bones together. If those are bad, then the hips are not going to function over time, and we're going to see dysplasia develop. And so any one of these, or a combination of these, can all produce the same condition of hip dysplasia. And the genetics behind this are all over the place. We're looking at multiple genes controlling each one of these etiologies. Now, the interesting thing is that genes have two roles. Most of the time, we think of genes as something as this gene produces this protein, which is used in this physiologic process. But genes can also have a regulatory function, which means that this gene may not produce the protein, but it controls the expression of another gene, which may or may not have an issue. So it almost complicates it, by an order of by 50% or more, because now we're not only looking at the genes that do something, but now we need to look at the genes that control or turn on and off the other genes which do something. Now, the significance of everything that we understand of the genetics of hip dysplasia is currently very vague, as which you're all aware of because we don't have a genetic test for it. And a part of the reason why that information is vague is because it's very difficult in science to replicate or consistently produce findings as they are published. And this has to do with the variability of the phenotypes, meaning we got that 40-fold difference in dogs from the smallest to the biggest. And so it's very hard to control all the variables. And at the same time, a lot of times we can't get the adequate sample sizes when that's required to produce truly significant results in our scientific studies. So in 2021, the researchers produced a paper which was very, very interesting when it comes to hip dysplasia. And was made part, in fact, by the fact that we can do so much commercial genetic testing. There became a much more available data set that they could use to look specifically at this issue. Now, the point of the paper was to validate 46 different known genetic markers from previous research papers, and they used a cohort of nearly 1,600 dogs from 10 different breeds. So this was the largest study cohort to date. And the idea is they wanted to look at those 46 genetic markers that had come from previous publications and said, if we take these markers and look at across 1,600 dogs, how many of them are truly going to show valid, are truly going to be valid for their role or their association with canine hip dysplasia? And what they found is they found 21 of those 46 were actually significant, statistically significant. And these were spread across 14 different chromosomes. Now, 20 of these associated with canine hip dysplasia were very specific to the breed, whereas one was ubiquitous across all the breeds. Now, this further characterization of the associated loci could reveal that canine hip dysplasia relevant genes and pathways, so that they could improve their understanding of how the disease actually develops. So this was a really interesting study because now they could take what we've learned what we learned previously and say now we have a much more focused pathway to looking at these 21 different loci and say how do they actually affect canine hip dysplasia. Unfortunately, environment still plays a role. We all know that calorie intake, lifestyle, level of exercise, and diet both supplement the dog's body weight and muscle mass, and those directly contribute to whether or not a dog could have bad hips. Now, because fundamentally bad hips form arthritis, and we know that arthritis comes from either normal forces on an abnormal joint, something in the joint isn't right, but we can also see if there are abnormal forces on the normal joint, we can also see the development of arthritis. And when we're talking about obese or overweight dogs, they produce abnormal forces even though their joints are normal. The other point I want to make here is that there may be distinct, which means separate, but related genetic influences. And that's going to lead us to our topic about canine obesity. Okay, I'm bringing back the ugly words again. Here we're talking about monogenetic versus polygenic. Now, I want to show some examples of a monogenic study and a polygenetic study to show you just what we understand about canine obesity. All right. Now, before I get into those, I want to introduce you to a bad apple, an interesting apple. I don't want to call it bad just yet because we don't know a lot about it. These are structural variants. Now, structural variants could be the key to unlocking many polygenetic traits, but they could be the key, but are they also the lock? Now, what is a structural variant? Structural variant is a region of DNA that is very large in size, and it can include diverse mutations, making the detection of an individual section of DNA very complicated. Now, structural variants can actually be, and they are, the major source of genomic diversity. They're the reason why we have such diverse and unique breeds, but their functional effects are not well understood. So when it comes to using a genetic test to determine the presence or absence of a structural variant, it's very, very complicated. It's like, with the image here, it's like trying to find one thing in that closet full of things. You want to find your blue shirt, it's in the hallway closet. You open up the hallway closet and you see this. Now, you can find the blue shirt, but it's going to take you time and it's going to take you effort. And oftentimes you're going to decide, you know what, I'm just going to go with the green one that's right over here because it's just not worth all that effort. That's kind of where structural variants are right now. We can detect them, but it's not simple, it's not efficient, and it's not cost effective. All right. Now, you guys are like, what is he talking about? He's talking about monogenic, polygenic, he's talking about structural variants. Where is he going with this? You may be familiar or have had to deal with a structural variant in your breeding program. The Brindle mutation, which is associated with the K locus, is effectively a type of structural variant. Now, functionally, we know it allows dogs to express more than one color loci and the characteristic brindle pattern. But what's frustrating is that we can't do it or we don't do a genetic test for it because oftentimes when we test these, we can't tell exactly where it falls between KB and KY. And at that point, then, in order to truly find it, we would have to send it off for special processing, which really isn't cost effective for most people. Maybe someday, now with the advent of sequencing, a brindle test may not be that far down the way. But I'm using this as an example that you probably have been dealing with a structural variant at some point in time, and they may not be as unfamiliar as it sounds. All right, so let's back up a minute. Let's go back to the monogenetic trait that I mentioned earlier that has an influence in canine obesity. And we're talking about the POMC gene. Okay, this mutation is predicted to disrupt the production of two neuroactive peptides, beta melanocyte stimulating hormone and beta endorphin. So this mutation affects whether or not the body can produce these two peptides. These peptides have a known direct role in appetite stimulation. In both ways, they can turn appetite on and they can turn appetite off. And therefore, they have been associated with greater weight in dogs, adiposity, and food motivation in affected dogs. Now, I bolded the affected dogs because one study suggested that this was an additive effect, meaning a dog with one copy of this POMC gene mutation would show greater appetite and be a heavier, more obese dog than a dog that is clear of it. Later research shows that dogs have to have two copies of this mutation in order to show statistical significance of whether or not this gene is assumed. But here is an example of the fact is that we take a very complicated issue like canine obesity and we try to reduce it down to one single monogenic trait, we're going to run into issues and we're not going to get the whole picture. So let's expand our thinking. Now that you're comfortable with, or at least you've been introduced to things like polygenetic and structural variants, let's look at another paper that came out just recently that found 6 genes with structural variants, and each had a suspected role in promoting or preventing obesity. To go through these relatively quickly, and I apologize, the ALPL gene, now what we know about these genes has come from previous research that was done both in humans and other animals. And we know that the ALPL gene regulates energy balance and adiposity. So we know that this one gene in other species has been associated with 2 factors that can control or contribute to obesity. The KCTD8 gene plays a role in bitter and sweet tasting signaling pathways. So here we're seeing 2 etiologies, polygenic, that can both contribute to whether or not a dog becomes obese. One is in energy regulation and balance, and the other is, again, in food appreciation, food motivation. And we look at the SGSM1 gene, and this has been associated with insulin measurement. And now we're hearing more and more, as the media likes to talk about, that obesity and its relation to insulin, whether we are insulin sensitive or insulin resistant, has becoming much more commonly accepted, and we're finding out much more about it. And now we're seeing that there is a genetic undertone in which insulin is being affected by a gene or a mutation in a gene. The SLC12A6 gene has a known role in movement disorders. And so here it may have a regulatory function in that this may be something that prevents obesity in normal dogs, but with the structural variant, now suddenly the dog is not as motivated to get up and get going and burning less calories. The RYR3 gene is suspected also to have a function in muscle contraction. So here's another etiology, movement in these last 2 genes are showing a direct association with something that can contribute to something that may prevent obesity. Now, as an interesting fact is the RYR3 gene is related to or in the same family as the RYR1 gene. Now, this gene is known to have an effect in meat quality and the fat level in pigs. And so we see that, this stands to reason that this family of genes has a significant role in canine obesity. And lastly, we have this VPS26C gene, which was found consistently. So it was statistically relevant. But at this point in time, we don't completely understand or know its function and how it rolls, which means, again, we have a lot to learn. So here we have 6 different genes that all have very different effects on the body. Each one potentially could contribute to an overall presentation of overweight or obese dogs. All right, so let's bring this all together. Now, as we transition to sequence-based testing, it may become significantly more possible to reliably detect structural variants in an efficient and cost-effective manner, which means that we may be able to start bringing these tests available to the consumer, which means that we might have the ability to start doing some really, really interesting things. And then as we start to do more and more sequencing of more and more dogs, we're going to be able to go back and the ability to detect and to start to make associations between things that we find in the genome and the phenotype of these dogs. We might be able to start bringing these together in a much more consistent and regular manner, which means that the future of testing is going to get very, very interesting. Now, when we're testing for complex traits, we know that these polygenic mutations that we're testing for are effectively going to becoming risk scores. They aren't going to be your dog has this gene, therefore it will do what. It's going to be your dog has these genes, and we suspect that its risk of developing this condition is going to be X. And that is because we're putting multiple pieces together. It's going to basically allow us to see what is the potential risk of an animal. Now, whenever we talk about risk, we talk about prevention, which means that if we understand that a dog is at risk for something, then we can take active steps to prevent it. And what's interesting when we're talking about obesity, particularly, this is one of the most difficult conversations a veterinarian is going to have with a client for a lot of different reasons. Nobody wants to hear that their pet is overweight. And the other thing in our society, food equals love. People are very sensitive on what they feed their dogs and how they feed their dogs. And they don't want to be told, you might be loving your dog a little too much. But if we have the genetic basis to say, you know what, your dog is at higher risk for obesity because of this gene, and this gene, it may be the template that a veterinarian can use so that we can start to talk with pet owners more consistently and regularly and actually affect how obesity works with these pets. And then finally, as we develop more and more of these tests, we're going to be able to improve the overall health of any of these effective breeds on both levels, both from a breeding standpoint, meaning that we can breed to reduce the risk, and at the same time, is even though we know that there's a risk there, people can go into this forewarned and forearmed to impact the individual pet's life. Now, just to summarize, again, I threw a lot of science at you in a very short period of time, and I apologize. We did talk about the single nucleotide polymorphism. That's the SNP. That's the basis of testing technology today. And we're transitioning to sequencing, where we're going to be looking at all the base pairs that make up the sequence within the DNA. We compared and contrasted monogenic versus polygenic traits. Monogenic, one gene, one trait, versus polygenic, which we're looking at a number of different genes, all contributing to a problem in a number of different ways. And we used hip dysplasia and obesity as an example of that, finally, so that we can hopefully increase our understanding of genomics. Now, I often like to say is that one of the frustrations when I give these presentations is that I do a lot of, we're going to get there, we're going to do this, we don't completely understand that. And it feels a little like I'm almost a broken record when it comes to genetic testing. But I always think that we are just one discovery away from really changing the way we produce dogs and the way that we impact their health and improving their quality of life. So with that, I just thank you very much for listening. And this is going to be recorded, so you're going to have access to my contact information. I know genetics can be complicated, and so I'm always welcoming. If you want to send me an email, I would love to respond to your questions. These were just some of the references that I wanted to give proper respect to.
Next presentation, we're supposed to have a message now from Mari. There she is.
Yes. Thank you very much, Will, and thank you, Dr. Wester. That was very interesting. Just want to give everyone a little bit of information on Neogen's paw print genetics. Paw print genetics by Neogen is a selection of nearly 350 breed-specific tests. We offer one of the largest testing portfolios available, providing valuable genetic insights into diseases, risk factors, traits, coat colors, and more. We provide breed-specific panels and individual tests to help enable relevant testing for your breed. Now we have another genetic test that's a comprehensive screen called Canine Health Check. So Canine Health Check is a comprehensive genetic screen for any breed of dog with over 325 disease-causing mutations, traits, and coat colors. You can uncover useful information such as mutations associated with various diseases, color trait genotypes, and more. Our analysis classifies results into clear, carrier, or at-risk affected categories, providing a holistic understanding of your dog's genetic health. Neogen, we are the largest animal genetics lab in the world, and we are striving to make the technology of the future available to you today. So thank you very much, and I'll pass it back over to Will.
Thanks, Mari. Well, now we're right on to our next presenter, and we all know Dr. Marty Greer. There she is. Hi, doctor.
Hi.
Floor is yours.
Wow. All right. It's really hard to follow a presentation like Dr. Westra. He's amazing. So glad that we could all participate in that. I do have some things that kind of correspond to what he's talking about. Oh, we got to go back a little bit. Sorry. I was messing around with my presentation, of course, because I'm not done writing it until we start. So here we go. So we're going to talk tonight about the challenges and joys of breeding our large breed dogs, because there is a lot of interesting differences between those and the small breed dogs. Some people have a combination of those breeds. Some people are just large breed people. So we're going to talk about those. I started off with Clifford, the quintessential big red dog, because I thought he was kind of fun to start with. So we're going to talk about large breeds. So the challenges are really the things we're going to cover tonight. We're going to talk about best practices during pregnancy, nutrition during pregnancy, colostrum, pyometra, and then just some miscellaneous comments that we did a little brainstorming here at the practice with the other doctors, coming up with some of the other things that make different breeding a large breed dog than a small breed dog. So we're going to go through best practices during pregnancy, keeping our dogs fit, swimming, bathing, scheduled C-sections, parasite control. We'll talk about nutrition, basic body condition, basic concepts of nutrition, deficits, appetite, and water consumption. Then colostrum, because there's some kind of interesting things there. Pyometra risks, which I hope I come up with some things for you tonight that you go, wow, I had no idea. And then some miscellaneous items. So best practices during pregnancy. It's important that during our pregnancies that we keep our females fit. We want to keep her in good body condition. We don't want her overweight. And that can be sometimes a little bit difficult. We don't want to overwork her. We don't want to have her out in the field doing work. If she's a field dog or a performance dog where we have her overheated, we don't want her tromping around too hard on her body when she's in late-term pregnancy. But at the same time, we need to keep her fit so that she's in a good physical condition when she goes into labor. And we also don't want her to lose her mind, because sometimes keeping these dogs that are really used to being active the end of pregnancy can be a little bit difficult because they do get a little bit bored, a little bit wiggy. And of course, we want to keep her mentally challenged as well. Swimming, I get a lot of questions about swimming in Wisconsin. We have a lot of dogs that are field dogs, and they oftentimes do go into the water. I do recommend that you limit swimming toward the very end of pregnancy for two reasons. It's a great exercise, but during late-term pregnancy, we don't want any water getting up into the cervix after the cervix is opened, allowing an infection to ascend up through the cervix and into the uterus. I also don't want you to have to dive into the water and pull your pregnant dog out, because her body composition changed to the point that she sinks like a rock when she's late pregnant, and she may not have the lung capacity because of the pressure of the pregnancy to get herself back out of the water. So be a little careful with that. We don't want her going glug, glug, glug. And then one of the things that I can't overemphasize enough is the importance of having our bitches, regardless of their size, small breed or large breed, bathed or ready to go and nice and clean before she whelps or before she has a scheduled C-section. I'm constantly amazed by the number of dogs that come in that are not really as clean as we'd like them to be at the breeding, not as clean as they should be at their pregnancy and C-section timed end of pregnancy. So I think it's really important that we consider the importance of that. I don't want her going into labor when she's damp. I want her to have some of the oils on her mammary glands that allow the puppies to find her when they're born. But 3, 4, 5 days before she's due to go into labor, give her a good, nice bath. Get her cleaned up. If she needs her tail wrapped, if she needs her feathers trimmed, if you need to change the hair coat a little bit on her belly so the puppies can find the nipples and not get lost in big dreadlocks of hair, make sure that she's cleaned up and ready to go to surgery if that becomes an imperative portion of her labor and delivery. I do want to mention scheduled C-sections, and I think these are really important. They do pertain to litter size. Many of our large breed dogs have bigger litters than our small breed dogs, as you would expect. I mean, we do see Cavaliers that have 11 puppies, but that's not the norm. But we do see large breed dogs oftentimes having 10 or more puppies. So a bigger litter can mean a couple of things. One is the more puppies that a bitch has, the more likely she is to wear out, become tired during labor and delivery. The longer the uterus contracts, the more chance that this female is going to have that she loses puppies during the later parts of labor because of the number of times that the uterus contracts, the disruption of the blood flow to the placentas, the long distance that those puppies have to travel if they have to go all the way from the last rib down to the cervix to be delivered. So I always want to think about whether we want to schedule a C-section, and we'll talk about that in a minute. The other question is, do we want bigger puppies? Do we want to have our large breed dogs to have 4 really big puppies or 12 really little puppies? So larger puppies aren't always better. They do have better survival once they're born because they do come out with a better advantage. But on the other hand, it's harder for a female to deliver those 4 really large puppies because they are so big. So it does increase our risks. And of course, uterine inertia, where the uterus just wears out, is more likely when we have larger litters. Now, there's 2 kinds of uterine inertia. There's primary and secondary. Primary is where they just don't ever go into labor and don't really have effective labor patterns. And we see those in Bernese Mountain Dogs. We see them in Greater Swiss Mountain Dogs. We see them in a few breeds like that. We have secondary uterine inertia, which is just exhaustion. And it's not hard to imagine that after you've delivered 12 or 13 or 14 puppies, that you're exhausted, the bitch is exhausted, the uterus is exhausted. So it can be difficult to get those puppies out. So this is a picture of our dear Ruby. Ruby, on her 7th birthday, was raising a litter of 15 puppies. She came to the practice the day before Christmas a couple of years ago, belonged to one of my clients, and she came in for her puppy count x-ray on Christmas Eve. My staff member met me at the back door and he said, "Your first appointment is here." And she's crying. I'm like, "Oh, great. Christmas Eve, first appointment of the morning. Great. Let's start off the day that way." And I said, "Why is she crying?" And he said, she looked at the x-ray. She said there are too many. Now, this female previously had 6 puppies and lost 3 of them. So the owner understandably really concerned about what was going to happen with the larger litter. So we merrily went on our way and scheduled her for a C-section the day after Christmas. And she had 15 puppies, raised 15 puppies, and did a fabulous job. Here's our litter of 11 Cavalier puppies. Yes, all one litter, no kidding. So what do we do with a planned C-section versus an unplanned C-section? So C-sections can be planned, and we do base that planning on progesterone timing. We can't go into all the detail today about progesterone, but we'll leave it to say that progesterone rises at the time of ovulation. Typically, we consider ovulation to be when the progesterone is around 5, depending on the lab, depending on the machine, somewhere between 4 and 8. And pregnancy is 63 days long from ovulation. Now, it can be a 24 to 48 hours swing either direction, but we can really close in pretty tightly on this. I had 3 C-sections over the weekend, and yeah, we knew exactly when they were all supposed to have their puppies within a 24 to 48-hour period. So that's good news, is we can really time these effectively. So why do I have a push me pull you on the screen? To remind you that progesterone timing isn't just for the front end of the pregnancy, not just to tell us when to do the breeding, but it also tells us when the puppies are due to be born by C-section, be due to be born at all. And if our bitches aren't going into labor on time, that we should do an intervention before the placentas start to degrade, because pregnancy is really meant to be 63 days plus or minus 48 hours. So it's really important that progesterone testing is taking place. Many of you may say, well, I know I don't need it to get my female pregnant, but you may need it to get her unpregnant. So remember, the reasons that we do progesterone testing aren't just for breeding. Now, puppy count x-rays, in my humble opinion, are probably the second most important thing that you can do, secondary to timing your breeding correctly with good semen, to having successful outcomes of pregnancy. So I think it's really important that you consider this. If you're currently not getting good x-rays, I'm going to give you a few tips for you to take back to your veterinarians. Hopefully there's some veterinary team members on the call, veterinarians and veterinary staff. But I'm going to give you a couple of tips that you can take back so that you can get more effective, accurate puppy count x-rays. One of them is demonstrated in this picture. We take 2 x-rays. We take one with the female's side right side down and one with the female's left side down. And that makes a big difference. The other things that we do are we have our females come into the practice when we're taking our puppy count x-rays fasting, so no breakfast. If you can schedule a morning appointment, of course, that's nicer for your female so she's not famished by the time we get done. But food in the stomach can really significantly interfere with our ability to accurately count puppies because it overlays the uterus and makes that puppy count very difficult. Number 2 is matching or using a suppository. And for people that are familiar with this, you're just going to shrug and say, yeah, we already know that. But it works really well for TCIs and it works really well for pregnancy count x-rays. So either putting in a match, and yes, I really do mean a match, and I mean paper matches, please, not wooden matches, or putting in a glycerin suppository. Either one of those can be pretty effective to getting the female to pass a stool so that we have a nice empty colon. So if my staff puts a dog on the table, takes the first x-ray, and they can see that there's too much fecal material in the large intestine, they are trained to not even take the second x-ray. They are trained to put in a match, hand the leash off to the owner, and send her outside. Now, if it's June, they're probably going to take the dog out by themselves. If it's January, they're probably going to give that leash to the client and say, have a good time out there walking your dog in the snowbanks. So it's important that we have these stools out of the way so that it's a really good clear x-ray. Like I said, we take a right and a lateral. I don't take them with them on their back because the backbone overlays the fetuses and it can be very difficult to count. So I take a right side down and a left side down. Digital x-rays, of course, help. And almost every vet clinic at this point is taking x-rays digitally. And after day 55 of pregnancy makes a big difference. If we do it much before that, then we have a lot of immature skeletal development and it's really difficult for us to see the puppies accurately. So those 5 things can make a big difference in getting accurate puppy count x-rays. Please, please, please do not put your x-rays on Facebook and ask your friends to count. If your vet can't count them, by the time that we take that x-ray, which is in a DICOM image format, and grade it to a JPEG image, which will then show up on Facebook, the quality of the x-ray is so diminished that there is no way that anybody's going to accurately count unless there's one puppy on the x-ray. So please go to a veterinarian that can accurately count your puppies or a vet tech that can actually count your puppies, and do not count on your friends on Facebook to get you accurate counts. And as we know, Dr. Westra significantly talked about the differences in breeds, sizes, and how variable our dogs are. You can really look at how big of a difference that we have. So obviously, birth weight makes a big difference in how effectively these puppies are growing up. If we have puppies that are very small puppies and not thriving, it can be a lot more difficult for us to raise these guys. We know puppies in the lower 25% of their birth weight range for their breed are at increased risk of early death. So again, we want our puppies to be as big and robust as possible and still come out from the female either by C-section or by vaginal birth. So birth weight does have a strong relationship to breed. Parasite control, I just kind of feel obligated to throw this in here. I think it's important that we discuss it. Now, you in the northern parts of the world don't have the parasite issues that we do have in the US, particularly in the more southern states. But if you have a female that came from a southern tier state somewhere in the US or from other countries that may have a parasite problem, it's really important that we do talk about parasite control. And this is not specific to large breed dogs, although I will say more large breed dogs have parasites than small breed dogs simply because they have differences in how much time they spend outside and their exposure to parasite type of environments. But panax-fenbendazole can safely be used in the pregnant dog to prevent the migration of roundworms and hookworms that can happen through the placenta or the mammary glands. So giving this for the last 3 weeks of the pregnancy through the first 2 weeks of lactation, so for 4 to 5 weeks total, can make a huge difference in the number of parasites that our puppies have. And of course, puppies with parasites aren't thrifty. Puppies with parasites don't grow well. They have diarrhea. They don't feel good. So it's really important that we're doing a good job with parasite control, particularly if these dogs are from the US or other parts of the world where they might have parasites of concern. This is a very safe protocol. It's not one that is frequently known about by veterinarians outside the group of people who do a lot of canine reproduction, but this was information published back in the early 1980s, 1981, 1982, 1983. These articles were published. So if you're looking for these articles, please don't hesitate to email, and I will be more than happy to send them. And you can share them with your veterinarian, or you can actually Google these, go to Google Scholar and look up bendazole pregnant dog, and you will find references to these articles back from 40 some years ago. So not new information, but sometimes not adequately circulated. The dose is the standard dose, 50 milligrams per kilogram once a day for the total of 4 to 5 weeks, the last 3 to 2 weeks of the pregnancy. So starting on day 42, going till the days that the puppies are 14 days old. So please, 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 if you have parasites, if you have Giardia, roundworms, hookworms, any of those parasites, this is a really effective way to manage those. Okay, the second thing I want to talk about is nutrition during pregnancy. And body condition is really important. Ideal body condition is where we want our bitches to go into their pregnancy, or maybe just slightly heavy. Now, there's different scales that we can use for measuring body condition. A lot of people use a 1 to 5, other people use a 1 to 9, but the ideal body condition on a 1 to 9 scale is between 4 and 5, where you can feel the ribs, but you cannot visibly see them from across the room. Now, if you have a whippet, it's a little bit more of a challenge to have those ribs not show. But the average dog, you should be able to easily feel the ribs. So if you take your hand and make it into a fist and you run your fingers across your knuckles, the ribs should feel like the back of your, like the back of your hand. If you can feel like your knuckles, she's too thin. If it feels like the ball of your hand, she's too fat. So the back of the hand is what you want her ribs to feel like. You can teach this to your partner, your spouse, your kennel help, whatever, but we want the ribs to feel like this. It's okay if they're just a slightly bit heavier than that when they go into a pregnancy. I don't want them at fighting weight if they're performance dogs. I want them just a little bit heavy, but certainly not fat, not obese. We know that obesity interferes with good pregnancy. It certainly interferes with effective labor and delivery. So we want them to be a little soft, but not fat in any stretch of the imagination. Now, a lot of people have been feeding raw meat diets. We see more and more all the time. We also had a period of time there, we were seeing a lot of carbohydrate diets. We're still seeing some of that, but not quite as much as we did before. So I think it's important that you understand that raw meat diets can create some significant issues during pregnancy with bacteria and parasites. Carbohydrate-free diets can also create issues. We also can see calcium deficiencies associated with these raw meat diets or the unbalanced diets. And then, of course, we're going to talk about folic acid and DHA. So in general, we need to maximize our success rate by feeding our stud dogs, our bitches, and our puppies the correct diet. And unless we manage their health, nutrition, and environment carefully, we cannot expect these bitches to produce nice litters. We can't expect the dogs to produce sperm of good quality, and we can't expect our puppies to thrive. So I really talk about feeding the machine that feeds the puppies. And I hate to call it a machine, but really it's kind of the way to look at it is that if we don't put in the right caloric intake, if we don't put in the right nutrients, we're not going to get out the product that we're looking for. Garbage in, garbage out is kind of how it comes down to. So the basic nutrition is really important. And when you're looking at different types of dog food, you can look at the label, you can compare the macronutrients, which are fat, carbohydrate, protein, and fiber. It's easy to look at those on the label and assess what those foods look like. And most people assess what their dog's diet is doing for their dog by looking at their coat and by looking at their stool quality. But they forget that fertility is heavily influenced by these micronutrients that you can't really read on the label. So our recommendation is to use the Sport 30-20 diet or the large breed puppy food as our puppies start to grow, so that we really have nutrient profiles that we need. And we do have people that are like, well, I'm not really crazy about this kind of food. And you look at it and you say, well, look at what the quality of what your outcome is. And I'm going to tell you that when we have issues with people that are having fertility problems, the first 3 questions I ask them are, what do you feed? What else do you feed? Because many of you are feeding something along with the diet. You're supplementing with a home cooked food or with a raw meat or with something else. And then what supplements are they on? And I think it's really important that you look at this, because when we have issues with nutrition, we see it reflected as poor fertility. And as soon as we switch to the diets to different products, many times, most times, we can see a significant improvement in fertility with that alone. So if you're taking the food and altering it, remember that the pet food company has spent millions and millions of dollars developing the food that's of the correct nutrient profile. So you need to stick to that and not alter it by adding too many other things, supplements.
Oh, there she is. There's Sarah. Here's Sarah
Yes.
Manzi from ProPlan.
Can everybody hear me?
We can hear you.
Okay, perfect. Hi, everyone. My name's Sarah Manzi. I'm with Purina ProPlan. And while we wait for Dr. Greer to be able to get back to her sound, I just wanted to take a moment to tell you a bit more about what ProPlan has to offer for large dog breeders. We have a lineup of veterinary recommended high protein formulas for large breeds, including new sensitive skin and stomach large breed salmon puppy formula, which is going to start hitting the shelves next month. Our large breed formulas contain glucosamine and EPA, and omega fatty 3 acid for joint health and mobility. Also really exciting that we can announce that we are launching 2 new joint care veterinary supplements that are going to hit the shelves this spring. They're going to be available in options for small, medium, and medium large breed. And that'll be coming this spring. It'll also be available for rebate as well as on breeder delivery. And then I don't know if Dr. Rear has the slides up. But if you're interested in trying ProPlan, the ProPub loyalty program for breeders offers great benefits, including getting $10 back on every large bag of ProPlan, their new online rebate. So no more mailing in barcodes for those of you who are familiar with our old rebate program. Delivery to select regions for large volume orders. We have really great puppy kits with a coupon for a small bag of ProPlan, 3 wet puppy food cans. New for this year, we've also added a free box of Fortiflora for each new puppy owner, as well as 1 for the breeder when you order your kits. To learn more, contact us or sign up online. Please visit purina.ca/proclub. Or if you opted in to hear from a ProPlan breeder representative when you signed up for this webinar, we'll be following up with you in the next couple of days. So look for that email coming soon.
Well, while we're doing this, maybe we can start with some questions for Dr. Westra. Dr. Westra, are you there?
Yep.
There he is.
Can you guys hear me?
I can hear you.
Okay. I was in the chat, just kind of stood this, and I started to answer some of the questions that were, I thought, more on my side of the fence than Dr. Greer's. But feel free to put, if you guys want to. I'm kind of scrolling through them here, and it's funny, one of these ones came up with it, and I guess I was really praying no one would ask this question, because I feel like I talk about this at every presentation, is the concern about intervertebral disc disease and how it relates genetically. The question was, what are the best ways to breed away from IVDD when 90% of the available population carry it? The simple answer is you can't. Don't bother. IVDD and our current genetic understanding is that this is a risk assessment. So there is a known risk if a dog carries the CH12 FGF4 mutation or insertion. I'll also shorten that down to CDDY. If they carry the CDDY mutation, we know that that dog is at risk. We know that the mutation is dominant, which means that even if they have 1 copy, there's a 50% chance the puppies will have it. But what we don't know is how that individual mutation is going to affect the individual dogs, meaning that not every dog with 1 or 2 copies of CDDY will develop intervertebral disc disease. And so it's the kind of thing, is because it is a risk at this time, and the frequency of the mutation is found, like in your situation, 90% of the available population. To go scorched earth and say, I'm only going to breed clear dogs, or only clear dogs free of this mutation be bred, you're effectively creating a genetic bottleneck, and that is only going to create or lose genetic diversity within your breed, which is going to create more problems than it's going to solve. Now, for those breeds in which there is a significant population of dogs that either have one copy or clear of it, sure, breed away from it. Let's just reduce a little bit of the badness. But for the dogs where it's found in such high frequency, unfortunately, the answer is we have to test, test, test. And eventually, as we breed dogs with one copy of the mutation and to other dogs with one copy of the mutation, we get a 25% chance of producing a clear dog. And then over time, we'll produce more one copy mutations and more clear dogs, which can produce more clear dogs. And so, like I like to say, is that we're going to have to walk away from this. We cannot run. I'll take another question. This is a great question. It says, how can we breed away from melanistic mask in a breed where solid colors are the only acceptable for registration? That's a doozy. Because the interesting thing is that if you have a solid black dog with mask, you're not going to know that it has mask. And so the only way you're going to know if the dog has it is if you test for it. And then simply you're just going to have to basically test the parents and test the offspring and then remove those dogs with mask from your breeding program. And hopefully it's not something where, and this I'm not entirely familiar with, but I don't know about the prevalence or the frequency of the mutation within the program or within the breed that you're talking about. But you're right, you can't see it, so you have to base it on a genetic test to determine whether or not the variant that causes mask is present or not.
There's a question here, Dr. Westra. Pen hip versus OFA hip. Is that one for you?
I can answer it.
Pen hip versus OFA hip, one or the other or both for trying to prevent hip issues in future puppies?
So both are a legitimate way of evaluating the hips in the parent dogs. They both have their pros and their cons. And I will also say when Dr. Greer has the ability to chime in here, I would defer to her wisdom because she probably deals with this a lot more as I've been away from clinical practice a bit. But the way I see it, especially because I work with OFA, is that they both can provide an accurate assessment, but they have distinct differences. OFA, you can do it on dogs much sooner in life. Pen hip, you can do on dogs much sooner in their life, and you get a quantified score, versus with OFA, it has to be done after the dogs are greater than 24 months, and you get a qualified or quantitative score. And so there are distinct advantages and disadvantages. There was a question in there about whether the dog should be sedate for the x-rays. From my understanding, in order to get quality radiographs to have the hips accurately assessed, the dogs should be under some kind of sedation or anesthesia, but Dr. Greer may have a different opinion of that. There's a question here about overbites and how long it would take to breed away from it. The thing about overbites is when we're talking about the dental arcade is that from the last time I checked, it's been a while, there were about 9 different genes discovered that play a role in dental development. When we're talking about parrot mouths, probably the easiest thing to do is to go back to what I call is more traditional or conservative breeding philosophy, and that is that if you breed like to like, you want to get more like. And so if you have a dog with an overbite, this may be a dog that you want to remove from your breeding population if it is possible, just because even though we understand that the dental development is polygenic, multiple genes involved, you know that the dog that is presenting with the overbite has the conformation of genes that is producing the phenotype that you don't want. And so I know that there are a lot of factors involved with whether or not you breed a dog, but I would definitely put that in the con category when you're evaluating all the pros and the cons.
Okay. I think I'm better now. Are we
Oh,
good?
you are.
Boom, you
Okay.
sound great.
Okay, good.
Excellent. Nice to have you back, Doctor.
Yeah, and I don't know what happened because I didn't do anything, but we are in the boonies, so sometimes Wi-Fi is less reliable than what we'd like it to be. So getting back to raw meat diets, we don't recommend those. We can see an increased risk of bacterial infections in the bitches and the puppies. We see increased risk of parasites and, of course, an increased risk of pyometra because of the bacteria in the bitches environment. So when I talk about raw meat diets, I don't really think I need to say much more than what this slide presents. I think it's like kissing a pig. I think it's kind of icky. Do I like my steak rare? Yes, I do. But I do like to think that our females should be eating a cooked meat diet, particularly when we're trying to get them pregnant, they are pregnant, and when they are nursing. The carbohydrates are a really important aspect because for a few years we saw a lot of the no carb diets and we saw a lot of problems with those. Some of the cardiac problems have pretty much been dispelled, but it was really interesting to me that we know from studies that we need carbohydrates for the bitches to grow their puppies. During pregnancy, we know it's important for them to produce milk. And I was just at the VMX meeting a couple of weeks ago, and when I have a chance to go to any of these meetings, I go to as many presentations on babies as I can. So I went to the baby presentations on baby horses, foals. I went to the presentation on calves, the baby cows. And there was a really interesting study that was done in cows. And of course, cows are important for milk production. That when they fed the cows more energy in their diet, not more fat, they didn't make the cows overweight, but when they fed a higher energy diet, that the colostrum quality was better for those babies. So I think it's really interesting that cows produce better colostrum on a high carb diet. So I think we can potentially look at that as a relationship in our dogs as well. Now, nutritional defects or deficits that we can see can cause midline defects, including cleft palates, cleft nostrils, open abdominal walls, open thoracic walls, spina bifida, and hypospadia. Cleft palates, you probably are all somewhat familiar with. That's an opening in the roof of the mouth. Cleft nostrils, we can see either one or both nostrils with clefts. They can be associated with a normal palate or a cleft palate. And then this is a Labrador puppy that was born with the trifecta of abdominal and midline defects. The puppy had spina bifida, that's the red dot between her shoulders. She had a cleft palate, as you can see in this photograph. And then on the photograph clear on the right-hand side, she had an open abdominal wall as well. So she had all three midline defects. Now, there are two kinds of abdominal wall defects that are seen in dogs and in humans. There's one called an omphalocele, which is right down the middle, right on the midline. And in that case, when the intestines are born with them being exposed or the chest cavity is open, then those organs are covered with a peritoneal sac. If it's slightly off the midline, it's called a gastroschisis. And those don't have peritoneum over them. Now, sometimes you can tell the difference, sometimes you can't. If your puppies are born at home that way, chances are you're not going to be able to save them. If they're born by C-section, you might be able to. So there is an important difference there. But gastroschisis, when it happens in humans, is more common in Caucasian teenage mothers who smoke and drink. It's usually slightly off to the right side, and it's also associated with an incomplete intestinal tract or no rectum. So when those puppies are born with open abdominal wall, off the midline, no peritoneum covering it. Now, most of our dogs are not teenage mothers that have been drinking and smoking, but nevertheless, it kind of tells you that even young people can have these defects, that we can't usually save those puppies, but we can sometimes save the ones with an open abdominal wall if it's right on the midline. Now, this is what an open thoracic wall looks like. There's no way to save this kind of puppy. There's not enough rib cage to go around. This puppy is destined to be euthanized or die fairly shortly after birth. I have some icky pictures, but I also have some kind of nice ones. This is a puppy that we were able to surgically correct his abdominal wall, and he was able to go ahead and go home with the owner and be raised. This is another interesting midline defect called hypospadia, where the underside of the prepuce is not complete. So the penis is there, but the puppy doesn't have the preputial covering that it should. So not as common as cleft palates, but still an abdominal or a midline defect that you should be familiar with in case this happens to one of your puppies. There is a relationship in some breeds with folic acid reducing the risk of these midline defects. Now, there's only one published study that I've been able to find. It came out of Poland in this journal published in 2013 that showed that in breeds like pugs and chihuahuas that are at increased risk of developing these cleft palates, these midline defects, that with adequate folic acid supplementation, that you could reduce the incidence of that by a significant amount, by half to 2/3. So if you have breeds that have a tendency to have midline defects like cleft palates, particularly the brachycephalics, the pugs, the Frenchies, sometimes other breeds as well, that there is a genetic component to it, so we can't completely eliminate it, but it can help to have this folic acid to reduce the risk of that by supplementing. Now, 5 milligrams a day is 12 capsules of 400 microgram. Most people aren't going to do that with their dogs, but if you have a small breed dog, some folic acid is better than none. It should be started at least 6 weeks before the female is bred. Also, midline defects, in addition to being folic acid related, they can be related to certain antibiotics, like antibiotics including trimethoprim sulfa and Albon during pregnancy, and it can also be related to excessive amounts of vitamin A in the diet. So again, more is not always better, so be careful when you're supplementing that you don't mess up what the pet food companies have put together for you. Additionally, DHA is a really interesting nutrient. It is now added to many puppy formulas, but not all. So if you're not feeding a Purina diet, take a look at the bag and see if DHA is one of the components that they're adding. But it has been shown that DHA has beneficial effects on the brain and retinal function. Now, this looks a little weird the way that the writing is because this is an article out of France. So there are some things in this blue section that look a little weird. But we know DHA added to the puppy formula can make our puppies smarter. It's really interesting. And they have gone on to take that information, and now DHA is added to human baby formula and important for brain and eye development in humans as well. So those are all important changes. Now, where did this information come from? Well, there was a study done with almost 5,900 puppies at Canine Companions for Independence, one of the service dog organizations, one that I have raised 6 puppies for. They did a huge study looking at 200 nutrients, and they determined that they saw an increase or a decrease in graduation rate when the bitches had their first litter. They had 50% graduation, all the way down to the time they had their 5th litter. They were down to graduating 25% of that bitch's litter from a program for service dogs. So they set about trying to figure out what was happening with these females. And what they determined was that DHA, which is a fatty acid, was depleted in these bitches' system by having these recurrent pregnancies, that it was all sucked out of her system. So by supplementing it back, we've been able to make our puppies smarter. And we know from this study done in 2012, published in the AVMA journal, that it's safe. It improves neurocognitive function, it improves memory, psychomotor function, retinal functions, and that there are no detrimental effects. There was an additional study that I don't show here that showed that. So I think it's a good thing to consider if your puppy food doesn't have DHA in it, that you start talking about supplementing that. It's also been shown to reduce the onset of senility in people. So write down DHA, because you might forget to go to the store and get some. All right, appetite during pregnancy. There are frequently bitches that around 3 times during their reproductive cycle, we see their appetite diminish. One is during their heat cycle. At the start of it or in the middle of it, some of these bitches don't eat very well. They're distracted by hormonal changes. Of course, the boys in the house are also affected if you have those. So it's not unusual to see that change. At around the 3rd to 5th week of pregnancy, we frequently see the appetite decrease or diminish. It's sort of like morning sickness. There's increased amounts of pepsid in the stomach. They don't feel very good. So many bitches will come in not eating very well at the time of their ultrasound, and that's sort of reason to celebrate, as long as it's not something serious like a pyometra. But it's not uncommon to see that appetite drop off. And then again at whelping, just before she goes into labor and right after for a day or two, many of these bitches don't eat very well. They may eat just before they go into labor and vomit. They may not eat very well because they don't want to leave their puppies for the first couple of days. So it's not unusual to see these girls being a little fussy. B vitamins can be helpful in stimulating the appetite, and sometimes you have to feed a variety of foods. You can take things like more palatable food, maybe scrambled eggs. You can top dress the food. There's some things that you can do with some of the canned products just to get the female feeling a little bit better, and just making her feel a little bit more interested in eating. So if you have to, dress it up a little bit for her, she's very busy with what's going on in her little belly. So go ahead and spoil her a little bit. Again, additionally, we want to talk about lactation and colostrum, because without good lactation for our bitches and good colostrum for our puppies, we're not going to have good, successful outcomes. We talked the other day about fading puppy syndrome. There is no such thing as a fading puppy syndrome. It's a description of a group of disorders that we see. And frequently, it's simply that the bitches didn't have enough milk, or the puppies didn't adequately find their way to the milk bar and get really started on good nursing. Some puppies are born really vigorous, ready to nurse. Other puppies need a little bit more help getting started, latching, and getting things going. So if you can get the bitches started to lactate early, like I said, a high energy diet can be very helpful for that. We can use metoclopramide, which is a prescription product, to improve the lactation. Metoclopramide helps the female to make milk. So that's a dose at 0.3 milligrams per kilogram every 8 hours or 3 times a day to improve lactation. That is a prescription product. Oxytocin is the drug that we use by injection to let down milk. So metoclopramide makes milk, oxytocin lets it down. They do two completely different things, but collaboratively, you can improve her lactation with doing that. There are some nutritional supplements on the market that contain fenugreek, like Oxymama, and then warm compresses can also help just to bring the blood flow to the mammary glands. Very simple to do that. Just put warm compresses on and then let the puppies start to nurse. Additionally, oxytocin does come as a nasal spray, which doesn't really help with milk letdown as much as it does with maternal skills. So if you have a female that's a little touchy about having her puppies nurse for the first couple of days, you can purchase those things online without a prescription. The other things that I have personally experienced having an improvement in helping with bitches lactating are things like bratwurst. And I don't know why bratwurst works. It doesn't have fenugreek in it. I've looked at the formulas. I don't really know why bratwurst bring in the milk, but hey, we're in Wisconsin, so if we can feed bratwurst, we're going to. Sweet potatoes, root vegetables, and oatmeal, other products like that are what human lactation nurses help their female patients when they're trying to breastfeed their babies. So why shouldn't we feed some oatmeal? Why shouldn't we feed a little bit of sweet potato? Those are all things that we can do to help our females improve their lactation. The sweet potatoes and the carbs in the oatmeal is probably that carbohydrate source that improves. I don't know in the bratwurst if it's the fat, if it's salty. I'm not quite sure why it works, but I fry them. I don't boil them. Of course, they don't get beer with it, but little dogs can have small amounts, bigger dogs can have a bratwurst 2 or 3 times a day for the first few days. And again, I learned this from a Newfoundland breeder. It works beautifully. It doesn't work on every dog, but I tell you, it works very well. And even my vegetarian clients have learned to cook up the bratwurst and feed them to their dogs so that they end up with better lactation. So they need the carbohydrates, we need that good colostrum, and we need our puppies effectively gaining weight from the very beginning. We used to say puppies could lose up to 10% of their body weight and still be okay, but we now know from the studies done at NeoCare in France that that's not the case, that if they lose more than 4% of their body weight, we have a pretty high neonatal failure rate. So we want those puppies coming out, we want them strong, we want them out, and we want them nursing as soon as possible. So feeding the babies is very important, both from a lactation perspective and at weaning. So it's important that we get these guys started. I wish I had such an organized feeding pattern at my house as these puppies all are alternating colors, but it is really fun to get these little kids started on food. So weaning can start as early as 2.5 to 3 weeks of age if necessary. I like them nursing longer if that's possible, but we don't always have that option if we have a female that's not producing milk, if we have a female that's not crazy about her puppies by the time they get teeth, or we have puppies that just are overwhelming the female with the amount of caloric intake that they need. Sometimes we do have to go ahead and get them started as early as 2.5 to 3 weeks on a gruel. I typically will mix some of the puppy food with some of the milk replacers and get them started until we can get them switched over to an appropriate puppy diet. One of the other things that I think we often forget about is water. Don't forget that as soon as the puppies are 2.5 to 3 weeks old, you can put a bowl of water down and they can start drinking. And one of the fun things that we like to do with some of these little kids is to give them something to keep them busy. So this little puppy here is showing you how to keep puppies out of trouble. She's merrily going along here with her muffin tin. I've got a ginger snap stuck to the bottom of it with a piece of peanut butter. So we're keeping this puppy busy in the exam room while we're having a conversation. But these kinds of puzzles are simple things that you can do with your young puppies when they're at home and they're starting to make you a little crazy because they're awfully busy when they're 6 weeks old and not quite ready to leave the home yet. So, like I said, super important that we're giving these adequately appropriate nutrition. So both options are suitable, whether you want to use the large breed puppy or wean onto the same diet that the bitch was eating, the 30-20 sport. They're both designed to support growth and reproduction. There are some differences in kibble size and some differences in the formulas, but either of those puppy diets are appropriate for our large breed puppies. Like I said, don't forget water. It's the most important nutrient. And sometimes we forget that our puppies can start drinking even before they're ready to start eating. So I use water bottles. I use chicken waters. I use a lot of different ways that we can introduce water to our puppies and not have them go in for a swim. We don't want our puppies falling in a bucket of water. So keep it in a shallow container. One of the last things I want to talk about, the thing we're going to emphasize quite a bit here, is what pyometras are. And I think pyometras are greatly feared by a lot of our breeder clients. Pyo literally means pus uterus. It does strike fear into the heart of a lot of our breeder clients. Many of us come into the clinic saying, please, please, please don't let her have a pyometra. It is pyo, not pyro. And it is not a verb. Bitches don't pyo. It is, they have a pyo. It's a noun. Pyoing is a verb. It doesn't really happen that way. So where do pyometras come from? It's a combination of hormones and bacterial infections. It can be related to cystic endometrial hyperplasia and other changes. So which comes first, the chicken or the egg? Well, we don't really know, but we know it takes that combination. This is a uterus with cystic endometrial hyperplasia, demonstrating that this female who was supposed to be bred surgically, we ended up spaying her at her surgical breeding because this is what the lining of her uterus looked like. And it should be smooth and shiny, not cobblestone and covered with these cysts. If your vet comes out and says at the surgical breeding, well, I found a couple of cysts and I popped them, you're probably okay. But a uterus that looks like this is never going to get pregnant. So we ended up saying that this bitch was done with her breeding career. And these are the changes that we see in the uterine lining, not to this degree, but this is why bitches over age 6 have a 33% lower chance of successful pregnancies because of the changes in the uterus due to the constant bombardment of progesterone during heat cycles. There are two general types of pyometra. There's an open cervix pyometra. This is a female that has vaginal discharge. She's on the surgery table. We're going to get ready to spay her because she was really sick with a fever. But this is what open cervix allows you to see, is that pus draining from the uterus. The closed cervix pyometras are a little bit more of a diagnostic challenge because they don't come in with vaginal discharge. So the difference between open and closed pyometras are the diagnosis is easier if it's open pyo. The prognosis is a little bit different, but you can still medically manage closed pyometras if they don't have a fever and if the cervix opens on medical management within a few hours of treatment starting. This is not a pyometra. This is a uterus that has resorption sites. So this is a C-section uterus. This is not a dog that should be spayed at C-section. We never recommend spaying at C-section. You can biopsy the uterus if you see this kind of change, but this is what resorptions look like at C-section. Culture them, sure. Biopsy them, absolutely. Do not spay them because many of these bitches will go on to have successful pregnancies, and we never, ever, ever want to spay at C-section because of the significant blood loss that occurs during the C-section. What isn't a pyometra, because there is some confusion on this, pyometras are not metritis. That's an infection in the uterus associated with a pregnancy. It's a postpartum complication. We can also see mucometras and hydrometras. Mucometra just means mucus in the uterus. Hydrometra means fluid in the uterus. They are not the same as a pyometra. Now, one of the things that you're probably going to be a little bit surprised about is that there's probably a genetic component to pyometra. There was a study done in 2012. Here is the study. So if you want to take a picture, we'll come back to that slide, showing the incidence of pyometra in different breeds of dogs. These are European dogs. This was all a Swedish study because they have a lot of dogs on insurance and they don't spay and neuter everything. So this shows the incidence of pyometra by frequency, the incidence of mammary tumor by frequency and breed, and then the incidence of having both pyometra and mammary tumor in the same dog. Bad luck. Bernese Mountain Dog takes the first place on this at 46% of these bitches. If they are not spayed by the time they're done with their breeding career, they end up with pyometras. Next is Great Dane, Leonberger, Rottweiler, Irish Wolfhound, Staffordshire Bull Terrier, then a Case Hound, then Bull Terriers, Bouviers, Newfies, Airedales, and then wrapping up the list of 12 is the Cavalier King Charles Spaniel. And when you look at this list, I see, and I'm hoping that you see the same thing that I do, is this large Mastiff type breed that was the predecessor, came from ancient Greece or wherever these dogs evolved in the European countries, that showed up as the predecessors to a lot of our giant breed dogs. So if you're guaranteeing in a contract that you are promising that you will guarantee the dog not to have a genetic disorder, be really careful what you say, because if you look at this and you realize that you have Bernese Mountain Dogs, Great Danes, or Leonbergers, you have a pretty high incidence of pyometras. Most people think of that as being a bacterial disease, but there's clearly a genetic component to it. Anytime you say, we see this more commonly in this breed than other breeds, you now have a genetic disease. Dr. Westra has really done a beautiful job of explaining some of this earlier, and there's probably a whole lot of genetic components that play a role in this, but we don't have a DNA test to say who's at risk for pyometra. So this is the study. If you want to go back and look at it, it's got the whole list of the breeds, but there's some breeds in there I don't see here in the US. There's some breeds in the US you don't see on the list, but it's a pretty interesting study. So you can click on this link, you can take a look at it and get the information if you'd like to investigate your breeds a little bit further. Pyometra symptoms are females that are sick 2 to 10 weeks after her heat cycle. Obviously, she's got to be intact. She's sick. She's frequently drinking more and urinating more, oftentimes vomiting, not eating well. They hardly ever run a fever, which is hard to wrap your head around, that they can have a uterus full of pus and not a fever. Some have vaginal discharge, some do not. Ultrasound is the best diagnostic tool. X-rays are not as good, but you do have to differentiate between a pyometra and an early pregnancy. So there are some differences. Progesterone testing doesn't really help you. White blood cell counts can be helpful, but both of these conditions, we can see a poor appetite and dogs that just aren't feeling well. Pyometra x-rays are very difficult to tell from early pregnancy. This is an example of one, but a pregnancy under 40 days can look exactly like a pyometra. So that's why ultrasound is so much more effective in determining whether it's a pregnancy or a pyometra. Pregnancies, we can see fetuses with little heartbeats, and pyometras we will not. So you can see on the right is a pregnancy, on the left is a pyometra. So treatment options do remain multiple for pyometras. You can get pregnancies successfully out of bitches that are medically managed with their pyometras. This is a dog that had a successful pregnancy. This is her litter. So you can successfully get these puppies, these females to have puppies if you don't spay them. This was my daughter's Bernese Mountain Dog litter. She had a pregnancy at, our pyometra at 2, a pregnancy at 2.5, and then another pregnancy when she was 7. So you can definitely get litters out of these females if you're successful at medically manage them, and if you have a veterinarian that's willing to do so. This is my daughter's litter of Berners. So I've been managing pyometras medically for about 22 years. Surgical complications of pyometra, if you decide not to manage her medically and instead to go to surgery, we can see peritonitis or uterine rupture. We can see the female have aspiration pneumonia. She can become septic. She can have kidney failure. She can have anesthetic death or go into DIC. This is what pyometras look like at surgery. This is not a pregnancy. You can see that they're segmental, but it doesn't look quite like a pregnancy. This is one of my favorite pictures. This is a Boston Terrier next to her pyometra uterus. You can see the uterus laid out is almost as long as the dog herself. Inside the uterus, there is pus, so it's pretty clear that this is a pyometra, pus in the uterus. So if you do decide that you want to try to talk about medical management, I always ask the questions of, are you planning to breed her again? If she over age 6, has she ever been bred? Because if she's never been bred and she's over age 6, you might want to spay her. If she has a history of being bred multiple times and never being successfully pregnant, you probably want to spay her. Is she healthy enough to go to surgery? How much will it cost to treat her? Know that there's a risk of recurrence, that you need to breed her back on our next heat cycle. I don't care if your son is getting married, you got to breed her back the next time. And if she's febrile, if she's running a fever, she should go to surgery and not have medical management. Medical management does require hospitalization, usually of 5 days or more. And every veterinarian that I know that's ever gone to vet school in the US has been told, never let the sun set on a pyometra. So these pyometra bitches are fragile, they're sick, they can really become sick very quickly. So if you do have a dog with a pyometra, you do want to either get her medically managed or spayed as soon as possible. What are the risks of management with medical treatment? Some of the drugs we use do cause some nausea, sometimes vomiting or diarrhea. We can see treatment failures where the uterus doesn't evacuate adequately. It costs about as much to do a medical management as a surgical management, so it's not cheap. It may sound less expensive because it isn't surgery, but it does require several days in the hospital and many, many injections of medication. She needs to be bred back on the next cycle. And if you have a vet clinic that's not familiar with or is unable or unwilling to medically manage her, you need to consider being referred to somebody that's able to do so. Medical management, we usually monitor the size of the uterus and look for it to reduce by 50% in size before we call the medically managed dog successful. And of course, the advantage is that you get puppies out of your medically managed dogs. Treatment in a nutshell consists of antibiotics, prostaglandins, which would either be luteolise or cloprostenol, and lots of walking, hand walking, because these females will have a lot of cramping if they don't get walked when they're getting their prostaglandin injections. We never send prostaglandins home with our clients because of the risk of humans handling the drugs, so we only use it in the hospital. If you're in Europe or some of the other countries where Alazine is an option for treatment, it's a great option. It's a lovely way to treat these dogs. They feel much better than they do with prostaglandins, but we can't get this product in the US and Canada. Some of the bitches need to be on IV fluids because they're vomiting, they're not feeling well. They may need Cerenia or metoclopramide for vomiting. They may need misoprostol to help open up the uterus. But if you're at an emergency clinic, if you can just get her on antibiotics and get her some supportive care with fluids and get her through the night, you can then talk to your regular veterinary clinic or find a referral place that will do medical management with a reproductive veterinarian and see if you can just get her through the night. At the end of a breeding career, we do recommend that you spay these girls because of the risk of pyometra, especially if you have those giant breed dogs that we're talking about tonight that are at increased risk of pyometra as they age. Up to 25% of all bitches can end up with pyometras at the time that they're age 8 and up. So any retired breeding bitch should be spayed. You may consider leaving her ovaries if you prefer to have her hormonally intact, but she should at least have her uterus removed so that you don't have to worry about pyometras. If you decide that you want to do a biopsy of the uterus at the time of your C-section, if you see those kinds of lesions, if she's got some fertility issues, we recommend using the Ohio State University, Dr. Premanand. He is double boarded in Theria, which is repro, and pathology. He's brilliant. So he does a fabulous job of reading these biopsies. Again, like Dr. Westra said, this will be recorded. So if you didn't get a photo, grab your camera. If you didn't get a photo of the slide, this will be up again for you so you can have your veterinarian contact them to submit samples. So some of the miscellaneous thoughts I have are what are the risks of breeding large breed dogs? We do see an increased risk of uterine torsion in those dogs. We had one this weekend where she had 0 puppies in one horn, 4 puppies in the other horn, and she must have laid down and rolled the wrong way. So her uterus had flipped. She wasn't able to deliver any of her puppies. Golden retrievers in particular, we can see high drops where an excessive amount of fluid accumulates in the uterus. We can see that in other breeds as well, but most frequently it's in golden retrievers. Lyme disease and other tick-borne diseases. That's a big deal in certain parts of the country, including where we are. We see a lot of females come to us for breeding. We do recommend that they're tested and found to be negative for Lyme disease and other tick-borne diseases prior to the breeding. And if they're not, we may delay that breeding until we've got her managed because we know that we can see these bitches that are walking around looking asymptomatic with Lyme disease, Anaplasmosis ehrlichia. During pregnancy, they're immunosuppressed. Those diseases flare up and we can see some pretty significant changes. And I was also familiar with some articles that show that retrievers, the Goldens and the Labs, are at increased risk of Lyme nephritis, getting really sick from Lyme disease, even dying from Lyme disease if they're Labradors or Golden Retrievers. And then Bernese Mountain Dogs, for some reason, we're going to pick on the Berners one more time, that they have a higher incidence of Lyme disease than other breeds of similar size, similar coat, in similar climates. That was a study done over in the European countries as well. So really watch your girls and make sure that they're negative for heartworm, Lyme disease, and other tick-borne diseases before you breed. Of course, we've talked about obesity. Usually it's our larger breed dogs that are more likely to become obese. We don't want to breed obese dogs because of reduced fertility, because of reduced ability to deliver puppies. If we have our large breed dogs, they are a little bit more likely to lay on their puppies. So you may want to put those pig rails in around the edges of your whelping box or some way that you can keep the puppies safe. And of course, one of the other things we kind of tossed around when we were talking about this is the larger the dog, the more expensive their medication because we dose on a per pound basis. So anytime we have larger dogs, we have an increased cost of medication. So just be aware that little dogs don't cost you quite as much. They have smaller litters, they have, less necessarily smaller puppies may mean you don't have as much profit in a litter, and profit's not a bad word, people. It's okay to make money when you sell puppies. It's okay to, not lose money every time you go to a show, but medications can cost a little bit more as well. So sorry about the internet challenges. I'm not sure what happened because I stayed where I thought I had good Wi-Fi. Thanks for hanging in there tonight. I've got the book, the Canine Reproduction and Neonatology book, if you're looking for more information. And then we have a little word from our sponsor.
All right, so here is some of our large breed formulas that I mentioned before. And then Dr. Gray, if you want to go to the next slide. And then here is the incredible puppy kits that we send to our Pro Club members. Dr. Greer, would you like to take a break or did you want to just jump right into questions?
I don't mind if we go straight to questions. If somebody needs to take a bio break, go for it.
Are we ready for questions then? Sounds great. Hi, everyone. Welcome back. Just a reminder, I would like to ask you a question. Make sure they stay in the general topic of what we discussed today and not be too specific up to your own dogs. First question, Wendy Kirk. Any ideas why Bernese are consistently having litters of 1 to 3 puppies when they should be having 8 to 10?
Well, Bernese do have a high risk of pyometra, which is associated with cystic endometrial hyperplasia and poor fertility. Sometimes it's too much inbreeding, so you need to be looking at your coefficient of inbreeding. That's a good place to be looking at your genetic testing. So there's definitely a number of different things that can happen. You need to look at nutrition. There's a lot of things that you need to look at. Semen quality on the boys. We just have a lot of fertility issues in Berners. The males have prostate disease by the time they're 3.5 instead of older dogs like most breeds. So there's a lot of challenges in breeding the Bernese Mountain Dog.
All right, Joanne Perrant, Borzoi girl, 2 times pregnant, then reabsorb. What are the possible causes?
And the list is very long. I'm happy to send you a list if you want to send me an email. But it's a very long list. It can be, again, too close to line breeding. It can be bacterial infection in the uterus. It can be that she's got some other health problems. It can be nutrition. And of course, if you're still having ongoing problems, a uterine biopsy is a pretty effective piece of information for your veterinarian to have. The other thing that we do with a lot of females that we are breeding now is we're using an anti-inflammatory like meloxicam on days 2, 3, and 4, and days 15, 16, and 17 post-ovulation. And we're seeing improved fertility with that. It seems to reduce inflammation in the uterus, and it's safe to use during those time periods. So I use meloxicam, Metacam, Mobec. There's a couple different names for it on those dates. So it's based on ovulation date, not breeding date. So days 2, 3, and 4, and days 15, 16, 17 after ovulation. And we do see some of those bitches that have previously been unsuccessful. There was a study that showed 50% of those bitches did get pregnant with that protocol.
Does that answer? Joanne also threw in, which supplement can we give to the stud to help fertility?
Let's start with good dog food. And then I do use some L-carnitine if we've got detached tails or bent tails. I do use International Canine Semen Bank CF Plus. That's their Canine Fertility Plus. Again, that's something you can buy without a prescription right from ICSB. So there's a couple of supplements that you can use that are successful.
Jill Miller writes, is there supplements you can give to the mom to increase the number of eggs she releases? Thoughts around breeder's edge problem female?
Nothing that I'm aware of on the market improves the number of eggs that are ovulated. I do have a part of my job is to work for Revival Animal Health, so I do have to have a disclaimer in there. So they do have some supplements, but again, you want to be careful that you don't over supplement and actually do damage to the nutritional balance that you have. And remember, when you look at the bag of the food, you're not looking at the ingredient list, you're looking at the nutrient profile. So don't be messing with that too much because there's a lot of important research that's gone into developing those diets.
And Julie Andrews asks, best treatment for mastitis?
Bathe before she goes into labor. Give her a good cleansing bath and use a probiotic. And that is probably the best 2 things that I can offer you to prevent mastitis.
Ashley Bartell writes, what causes short cycling in the bitch and how can we address this? Nothing on ultrasound to indicate abnormalities. Progesterone shows ovulation occurring 3 months heat cycle.
Yeah, some bitches do that and they simply don't get pregnant if they're not getting at least 4.5 months between heats. So your veterinarian needs to put her on some medication to extend the interestrus interval. Again, you can email me if your veterinarian wants some additional information.
And friend of the show, Kristen Musel asks, can an intact female develop pyometra if never having been in season, approximately 20 months old?
Never if she's not in season, but yes, if she hasn't been bred. So she has to have a heat cycle. By definition, a pyometra is after a heat cycle.
Julie Tong asks, is there a best option when tracking progesterone in-house, IDEX, Lab IDEX, or Lab True North?
I like IDEX test results a lot. They use the Imulite in their lab, and it's a very it's the gold standard for lab equipment is the Imulite. So that's what IDEX uses.
Alexandra Polakovich asks, is spirulina a good supplement for joint health and fertility?
I don't use that. I can't really speak to spirulina.
Superfood.
Carol Kokal asks, are silent heats signs of fertility issues?
Well, silent heats make it hard to find that she's pregnant, but that she's in heat. But you can get bitches pregnant that don't ever show a heat cycle. So you can do frequent vaginal swabs. You can get a stud dog to live with you. That can be pretty helpful because he'll be over there at her crate or at the door of the room that she's in, hollering, saying, "Hello, I'm ready to breed her." But you don't have to see evidence of a heat cycle to have the bitch ovulate and get pregnant.
And Alexander Pelikovich also asks, what joint supplements do you recommend for large breed dogs, and at what age should large breed puppies start on a joint supplement?
There's a lot of glucosamine and chondroitin products on the market that are pretty effective, but you need to make sure that they actually contain what they say. And some of the products, Purina has a JM diet, their joint mobility diet. There's a lot of diets on the market with pretty good joint profiles. So I probably think the most important thing is what Dr. Wester talked about earlier, and that's making sure that our dogs are not carrying excessive amounts of weight and that they're on an appropriate diet.
All right, Karen Watson, regarding the two types of in-house progesterone machines, Minivitis versus IDEX, is one better than the other? Do you have to read the numbers differently? What numbers are we looking for if using the Minivitis for a natural live cover breeding?
I gave up using a Minivitis 18 years ago. We had one, everybody's Minivitis has a different chart to interpret it with. So the veterinary clinic or the person that's running the lab samples needs to compare their Minivitis to a reference lab to know exactly what the numbers are. If you want to just know the numbers, just use the IDEX Catalyst. It's probably the best machine on the market for the in-house veterinary use. At in our practice, at one point we had 7 progesterone machines, so I can speak progesteronese. I can speak in that language because we ran them on many pieces of equipment. But the Catalyst is probably the most consistent, and you can't compare one hospital's Minivitis to another without a chart to know. You can't go online and use some other hospital's chart because it's not going to tell you accurate information. It also tends to drift if you're not rechecking those periodically. So I typically use the Imulite as our gold standard.
Annie DeRozier asks, do you recommend the pen hips test over the OFA test for hip dysplasia?
They're different. Like Dr. Westra said, the pen hip has the advantage of being able to do it younger. At 4 months of age, you can do it. It does require full anesthesia. And there is a correlation between the pen hip test telling you how much laxity there is in the joint and the development of hip dysplasia. However, OFA has a longer track record, and I don't think for some breeds that the hip laxity has the strongest correlation that pen hip would like us to believe. So I think we have to be careful how we interpret those results.
Andrew Younger asks, how to understand juvenile hip dysplasia? Is this condition always genetic, or do environmental factors still play a role even in a young dog?
It's both. As we discussed, we understand it to be genetically, it's polygenic. Multiple different genes are at play here. The study validated 21 different genes that have a strong association with dogs with hip dysplasia. But at the same time, the environment is just as important because lifestyle, activity level, nutrition, calorie intake, and body condition all play a role in whether or not a dog presents with hip dysplasia. So right at this point in time, we're looking at both. And then unfortunately, it's going to get even more complicated because as we start to develop those, better understand those structural variants and how they play into what I call distinct but related genetic conditions like obesity, now we're looking at 2 different aspects or 2 different pieces of the puzzle. So at this point in time, because we don't have significant or available genetic tests to deal with all of that stuff, let's control the things that we can control, and that being nutrition, lifestyle, and body condition.
Good footing under the puppies when they're really young. Activity, healthy activity for them so that they're not overdoing it, but they're getting a variety of surfaces. They showed a study that showed variety of surfaces help to strengthen the hip development. So there's a lot of things we can do when we can't control genetics, other than, of course, be smart about what dogs you put in your breeding program. Don't make excuses for putting bad dogs with bad hips in a breeding program. You're not going to get there fast if you continue to include those dogs.
Lou Carradine asks, any thoughts as to why Samoyeds seem to be so susceptible to A, packing on weight easily, B, developing diabetes, and C, bloating?
Yeah, I don't really have anything specific for that breed other than they're just the size they are.
Rita
Yeah.
Page asks, is there any situation where you would recommend an auto C-section versus allowing a mom to try to whelp naturally?
We do a
Okay.
lot of scheduled C-sections. Basically, it depends on the number of puppies. If there's 1 to 2 puppies or more than 9, we schedule a C-section. If she's a Bernese Mountain Dog, a Greater Swiss Mountain Dog, a pug, a Frenchie, a bulldog, we schedule C-sections. If she has a family history of difficult birth, then we do schedule a C-section. 75% of C-sections are caused by the bitch. So something on her side that caused her to have a dystocia. 25% are puppies, oversized puppies, puppies that are malformed, puppies that get in the wrong position, puppies that take a wrong turn when they come down one uterine horn. Instead of going out the cervix, they turn and go up the other uterine horn. So 25% of the time it's puppy, 75% it's mom. You can predict some of those, so we just schedule those C-sections automatically on day 62 after ovulation, unless they're brachycephalics. If they're pugs, Frenchies, or bulldogs, it's day 61 after ovulation. We just put them on the schedule, take them to surgery.
Alexandra Polakovich also asks, if a large breed dog is x-rayed at 6 months old and is free of HD and ED, then x-rayed again at 2 years old and is diagnosed with HD or ED, what is the likelihood that the diagnosis is environment-based rather than genetic?
You want to take that?
So if it's diagnosed earlier, I'm trying to sort here. If large,
It's at 6 months old, it's free of HD, then x-rayed at 2 years old and is diagnosed with.
no, because you can't really make that distinction because of the different etiologies that result in hip dysplasia. Some of them are very time dependent. So if it has to do with a laxity in the joint, meaning there's excess distance in there, as the dog gets older, it's going to be putting on more weight. That's going to become more pronounced. And then, of course, there is the time for that degeneration to occur that it would make the dog be able to be diagnosed. Dogs effectively that can be born with dysplastic hips, even though they don't look dysplastic at birth. I think that's part of the reason why OFA requires the dog to be 24 months before they'll take the x-ray, because they want to see a fully developed hip at that point to make any kind of assessment towards it. We see a lot of conditions that even though the mutation is there at birth, or basically at conception, it's one of those things where the environment has to kind of take time for that mutation to actually present clinically.
Yes,
Barbara
what he
Schrade
said.
asks, is pyometria a major risk when a dog has had only one scant heat cycle early in her life and has not had any more heat cycles?
No heat cycles, no pyometra, unless they're silent heats. So she's not at risk of pyometra if she's not cycling. But remember, dogs do not go through menopause. So she could continue to have heats even if you're not seeing them up until she passes away of old age.
Samurti Gunawardena asks, what might be the cause of primary uterine inertia given good history of fertility in the family and no concerns about fitness?
I don't know. It just sometimes happens. It just doesn't always go the way it's supposed to.
Nan Kennedy asks, is there a place where I can find a fertility specialist for my BCs. I have a 7-year-old female who has yet not settled. Have heard a course of antibiotics might help. Also, would like to have my male collected and his semen is checked for motility and possibly stored for future use. He's amazing.
So the theriogenology website, theriogenologists are the people who study canin or any animal species reproduction. There's a website, therio.org, therio.org. If you go to that and you go to find a vet, it will list all the veterinarians that are members of that society or college, and it'll be listed by zip code. You can put in a number of different filters by zip code, by species. So you put in canine, you put in what procedures you're looking for, and you can find a veterinarian by zip code in your area. I'm not good at helping people with it because the little towns that have names that I don't know that they're close to a big city, you have to do that yourself. So, but it's accessible. You don't need a password. You can get right into that website with no classified information. Just go in and take a look.
Samantha Keeler asks, after 55 days, counting from day of breeding or ovulation?
Ovulation.
Karen Watson, Dr. Greer mentioned Panacure for deworming. Can I use Safeguard 10% suspension?
Yes. Safeguard, Panacure, fenbendazole, those are all the same product. They're all, if you use the large animal product, use the suspension, don't use the paste, because the paste will not be uniformly distributed in the tube, but in the suspension, if you shake the bottle really, really well, it will be uniformly distributed and it's a nice product to use. It is large animal product, so technically that's off label, but a lot of people do it with good success.
Veronica Dollar, would running fecals on your bitch before or after be beneficial instead of deworming?
The problem with that is it only tells you what parasites she actively has in her intestines at the time. The bitches that ever had parasites when they were puppies will have those parasites insist in her muscles, and the stress of pregnancy and lactation will reactivate the migration of those. So you can deworm your female until you know what freezes over, and you can still have parasites in her muscles. So the only way to combat that is to use the fenbendazole during the pregnancy and lactation periods.
And John Durham asks, should the fenbendazole be given all at once or divided into 2 doses daily for the 4-week protocol?
Once a day, and the bitches hate it. They don't like the taste of it. You're going to have to argue with them every time you give it. You can mix it with peanut butter or something that tastes good, marshmallow cream, I don't care what, but it is absolutely worth it. I have people that come in and throw their arms around me and say, Dr. Greer, I've never had such a healthy litter of puppies. They don't get diarrhea, they don't feel puny, they're awesome. So it pays itself back in dividends.
Alexandra Polakovich, is 30-20 a balanced diet for weaning puppies, or is LB puppy a better option?
I think they're both effective, just depends really on the kibble size and what you prefer purchasing, what you have accessible to you. They're both good diets.
Beth Collins, looking for advice on excluding Goldens who fail one hip OFA, one elbow OFA, or have juvenile cataracts. Toss the baby out with the bathwater?
Those are hard ones.
Well,
Go
I guess,
ahead.
well, like I say, I guess we'll probably have 2 different perspectives on this. Is that, here you have to have a reason to breed the dog that has a negative or undesirable phenotype. And I think the thing is that as you continue to include those negative concerns, you're going to be dealing with them in your program. So again, I like the way Dr. Greer said it earlier. It's like, like you don't make an excuse for breeding a bad dog, and especially when it comes to juvenile cataracts.
So my personal take on it is if it's not life-threatening, life-altering, life-shortening, you can potentially include it in a breeding program. So there's some minor defects because there's no perfect dog that I think are acceptable to use in a breeding program. But as soon as you start talking about orthopedic problems, elbow dysplasia, hip dysplasia, or cataracts that can lead to a life-changing, I mean, dogs that can't see, they don't have good quality of life. Dogs that have painful arthritis don't have good quality of life. I would not recommend putting those in a breeding program. There are plenty of little problems that can go on that you can, there's lots of little things. You're not going to have a perfect dog, but get rid of the bad ones first because those orthopedic things that are life-changing should not be part of a breeding program, in my humble opinion.
Maja Menanen asks, should dogs be sedated for puppy count X-ray?
No, no. They just lay on their side.
Kara Marks asks, is it true pen hip measures the joint length or laxity, and OFA measures how well the ball fits in the socket? So both would give a more well-rendered overview insight of the hip conformation.
A lot of people do both. They will do pen hip to look for laxity, and that means when you put stresses on the joint to pull the knees together, how far out do the hips move? So you can see that comparison of with the hip pressed in and the hip pulled out, they will measure the difference. So yes, that will give you a different look at things than you will get when you do the OFA, which is basically looking at, is the hip socket adequately covering the acetabulum adequately covering the head of the femur, and you can manipulate that sometimes to make it look different, better or worse based on the positioning of the dog. So there's some play in there. I think both are really a good way to approach it. And once you've got the dog x-rayed for pen hip, you just send the OFA and you don't have to take extra x-rays because you've already done the OFA view for pen hip. But for pen hip, you haven't always done for pen hip, it's a different set of 3 views than you get for OFA.
At a different time.
Different time.
Dr. Nancy Coates asks, why Panicure, which dogs hate, 30 to 40 cc's for large dogs instead of more palatable Parental for the entire long worming protocol during PGN to day 14?
Because the Panicure the fenbendazole gets rid of that migration that you don't get with fenbendazole or with Pyrantel. Nemex and Strogen don't do the same thing as fenbendazole. They don't get that cystic stage that starts the migration again. So good question, different mechanisms of how the drugs act.
Michelle Drennan asks, I am an occupational therapist. I use Labrador retrievers in therapy sessions. Is it recommended to try to not allow the labs to eat raw meats? What about raw eggs?
Yeah, I don't like the raw meat because, like we said, there's a calcium imbalance, there's the parasites, the bacteria. Raw eggs, if fed in excessive quantities, you can end up with biotin deficiencies. So cooked eggs are fine, but you don't have, because you have to worry about biotin deficiency and salmonella in raw eggs. So I typically will say cooked eggs and cooked meat, please.
Shelley Millett asks, what is the cause when they are born with the top of their skull open on the back side of their head?
That's bad luck. I don't think there's enough of a genetic component to it. Is there anything that you know of?
No.
Okay, because we see those rarely. I've seen a couple of them in my career. It's horrifying. It's awful. But I don't think it's genetic. I think it's just stuff happens.
And when she says stuff, sometimes we look at it that the environment also includes the uterine environment. And so changes in the lifestyle while the dog is under development can have profound negative presentations that has nothing to do with the genetics at all.
Janice Waldo asks, what about penile feticulum? Is this also a midline issue?
I think she means the frenulum. And it is midline, technically, but it's because a membrane that developed didn't break down, and it causes the penis then when it is extruded and becomes either out for urination or out to be erect, that it pulls the penis down. So it's just a little membrane of tissue underneath that didn't break down. So all it has to do is be snipped and it's resolved. But it's not really a midline defect because it's not a gap, it's a failure for the tissue to recede.
Julie Summers, how do you feel about extra supplements like Breeder's Edge?
Well, I'm supposed to say I like them because I work for Revival. Um, to be honest, I use Oxymama, and that's really the only one I use on my dogs. I don't think they're
Elaine
bad.
Medill,
I
sorry.
just don't like to mess up the nutrition too much because we we go to a lot of lengths to get it right. So again, just be careful with over supplementing.
Elaine Medill asks, what milligram dose are you using for pregnant bitches and growing puppies?
For what product?
That's all we have.
Okay, if it's fenbendazole, it's 50 milligrams per kilogram. I'm not sure if it was another drug. There's a lot of drugs that we talked
Yeah.
about tonight.
Ashley Bartell again. What would cause a bitch to have zero milk colostrum even with a metoclopramide? Is this likely to reoccur with future pregnancies?
I think there's probably an inherited component to this because a lot of bitches, if they didn't lactate well, their daughters may not as well. So I don't really know. I mean, there's not, again, we don't have DNA tests for a lot of this stuff that we think has a genetic component to it, but certainly adequate nutrition, adequate water intake, all those things are really important. A lot of bitches don't drink very well at the time that they're in labor and early lactation. So they may be dehydrated and not lactating well. So giving fluids sub-Q, giving them salty foods to get them to eat and drink more, all those things can be helpful. And like I said, I don't know why bratwurst works, but if you haven't tried it, my husband was very excited the day I said, we're having puppies today. You need to go to the locker plant and get some brats. He's like, yes. I said, they're from Miranda. He was really disappointed.
Jill Miller asks or writes, I've heard a few times that feeding ProPlan can help with fertility. One thing I have trouble reconciling is if food can improve fertility, why is it that rescue dogs can often still have large litters despite having a poor diet? Trying to decide whether I should try to make the switch. My dog is very food sensitive and has been doing well physically on her current kibble, but has had one singleton and currently seems as though she might be having another small litter.
You have really very little to lose by changing the diet. So I would strongly recommend it. Part of the reason that we see great fertility in our rescue dogs is they're outcrossed. So we don't have to worry about them being genetically too closely line bred. God knows what they were bred to. And she may have been bred to more than one boy at the same heat cycle. So, we've got a lot going with that. And of course, it's just the way it is. Like you go to all the trouble to genetically screen your dogs and do all this stuff and feed them right, and then the neighbor dog has 12 puppies under the porch in a snowstorm and they all survive. It's just not, life isn't fair. We all know that.
True. Victoria Cash asked for Dr. Greer, since you touched on metritis, what is the ideal treatment course? Antibiotics, semi-urgent spay? What would that change if she was retired from breeding?
I really, really, really don't like spaying dogs when they are immediately postpartum or at C-section. Most of the time with metritis, we will ultrasound and make sure that there's not retained placenta or, of course, a retained fetus because that's really bad. So retained placentas are frequently the cause of metritis. So yes, antibiotics and then either oxytocin or a prostaglandin to help the uterus to contract and expel those components of the placenta that might be left behind. And there's some breeds like Bassets that seem to be at increased risk of developing metritis. Bitches that had difficult labor, bitches that might have been on progesterone to support the pregnancy, those all cause decreased uterine contractility. If she had a C-section, the placenta should be removed at the C-section and not left behind. So there's a few things that you can do. And again, probiotics. Probiotics came up a number of times a couple of years ago during the Repro meetings where it really seemed to prevent a lot of health problems. So getting the right bacteria in the system can make a huge difference. So really, I lean heavily on probiotics much more than I do antibiotics in those cases.
Veronica Dollar asks, okay, so if you breed a risk-affected dog to a clear dog, would this produce all carriers? What about the next generation? If you breed one to the carriers to another clear dog, is it possible to produce a clear, or is a carrier the best possible outcome?
So we have to be talking about a recessive mutation here, meaning it would take two copies of the mutation to produce that affected dog. Let's not talk about dominant. So to your question specifically, so you would produce all carriers. Now, if you breed a carrier to a clear, the clear dog can only pass on a clear gene. The carrier could pass on either the mutation or the clear gene. So you effectively have a 50-50 chance with that next generation of producing a clear dog or another carrier. But because the clear dog can only produce a clear gene, you're only at risk of producing another carrier 50% of the time. And that's a probability, right? You can roll the dice, 4 times in a row, it's going to come up a different time, or flip a coin 4 times in a row, it can come up heads all 4 times. It can come up heads, tails twice, or it can come up, heads once the time and tails the rest. But you have a 50-50 chance with that pairing that you proposed there to produce a clear or a carry.
Lindsay Yennon, what was the dose for Panacur for a pregnant bitch?
It's 50 milligrams per kilogram once a day from day 42 of pregnancy to day 14 of lactation.
Leah Foxcroft asks, I breed standard poodles and getting them to eat enough to keep getting them to eat enough to keep at a good, I think it's good weight, is challenging and more challenging during pregnancy. They are a pretty lean breed. Any suggestion?
That is a hard one because standard poodles are not hearty eaters. You should probably talk to Dr. Westra about finding the genetic marker to make them eat better. Most of the drugs that we use, like Entice, to improve appetite, we don't have any testing done on them during pregnancy. So really just trying to find the most palatable food. The 30-20 helps a lot because it is the higher fat content. But all you can do is find the food that she prefers the most. If she likes salmon, feed her a salmon-based diet. If she likes chicken, feed chicken-based. You just kind of have to do the best you can. Pepsid, famotidine, is safe during pregnancy. So if you do have a female that seems to be a little touchier with the way her tummy feels during pregnancy, you can use famotidine or Pepsid. But otherwise, you may just have to top dress the food with something delicious so that she'll keep eating. And you might have to give her some variety. I know it sounds like you're spoiling her, but if she's pregnant, she got to eat.
And to that point, is that one thing I thought, that came out when I was presenting is that a lot of the genetics that they're looking at for obesity are actually focused on the food motivation of the dog. This in the situations that I presented, they were using the Labrador, but it goes to show that there is a genetic component towards food motivation. And so that's going to present some interesting opportunities in the near future when we can start to look at some of those more complicated mutations to see just that it may be the kind of thing. Now, the ones that we were looking at were very breed specific, but we may find some of those more, ubiquitous genes in which, we can say your dog is going to be an eater.
Elaine Medill, sorry, my milligram dose question was about DHA.
Oh, good. DHA, we don't really have a published dose on it, but it comes as a capsule. So small dogs, I'd give one a day or one every other day. Big dogs, I might give one or two. We don't really have a milligram dose, but that's a great question. I wish we had a published dose on it.
Madeline Jacob, does the strength of the bleeding during the heat have an influence on fertilization? Madeline St. Bernard's.
No, the amount of vaginal discharge has nothing to do with when she ovulates or the quality of her heat cycle. It's just variable.
Marie Bouchard, with male dogs, what age is it advised to breed them till? Is it related to testicular cancer?
If you have a male dog that's still standing up and still making sperm, I think you should use them in a breeding program. That is an indicator of longevity, meaning better health, better temperament, less likely to have other health problems. I love using old male dogs in a breeding program. They may have a little bit of a decrease in their semen quality. They may need to have prostate support medications like finasteride. But, if you got a 16-year-old boy that can still manage to breed a female, go for it. That's what we want, is long-lived dogs. And there's no better test for being long-lived than being able to still mate when you're 16.
Sheila Standish asks, for Dr. Westra, what Neogen genetic panel do you recommend for a breeding female golden retriever?
Oh, go to the Paw Print Genetic website and the Golden Retriever Essential Panel. When we've organized the testing, we tried to do it by breed. And then of those mutations, we've evaluated them for a couple of different criteria as to what's been found to be associated with the breed. At the same time, what is the frequency within the breed? Because there are definitely some published mutations out there that, breeders have done a great job and removed effectively from the population. So even though they technically still exist, there's just not that very few dogs out there that may actually carry those mutations. So we try to promote and then kind of degrade tests out of the essential panel. So that's a good place to start. The other thing too is that you can go to your National Breed Club and talk about what mutations that they think are important. And then I know for the people within the U.S. or I don't know if Canada is the CHIC program also has some guidelines. And the way it kind of works is that breed clubs talk to genetic companies. It's kind of a circle of communication to try to promote which tests are relevant to which breed. So the answer does change from time to time, but the essential panel is a good place to start.
Victoria Rass asks, does Neogen cover MCT tests for the American bully?
MCT. I'd have to look into that.
All right. I'm afraid I think it's time to wrap it up. We've gone a little overboard, or over time, I mean. But it was a great session. Thank you. I want to thank Dr. Greer and Dr. Westra. It's a wonderful night. And to ProPlan and Paw Print Genetics by Neogen for making tonight possible. Thanks, everyone, and we'll see you next time.
Good night, everyone.
Good night.